everyone, welcome back to my channel. In this video lecture, we will cover a new chapter of biology, that is biotech and its application. So, biotechnology deals with industrial scale production of biopharmaceuticals and biological using genetically modified microbes, fungi, plants and animals. The main three critical research areas of biotechnology includes the providing the best catalyst in the form of improved organisms, usually microbes or a pure enzyme. Second, creating optimal condition through engineering for a catalyst to attack. And third, downstreaming the process technology to purify the protein or organic compound. Then the biotechnological applications in agriculture are agrochemical based agriculture, organic agriculture and genetically engineered crop based agriculture. Then plants, bacteria, fungi and animals whose genes have been altered by manipulating are called genetically modified organisms like GMO. And GM plants have many applications. They made crops more tolerant or abiotic stress. Then they reduce reliance on chemical pesticides and help to reduce post-harvest loss. And the application of biotech in production of pest-resistant plant example is the BT cotton, BT corn, BT rice and many more. The pest-resistant plant decreases the amount of pesticide used. BT toxin is produced by bacterium called Bacillus thuringiensis, and BT toxin gene have been cloned from the bacteria and been expressed in plants to provide resistance to insect without the need of insecticide and in effect created a biopesticide. BT cotton bacterium Bacillus thuringiensis provide proteins that kill certain insects like lepidopterans, coleopterans, beetles and flies mosquito. B. thuringiensis produce crystals that contain a toxin called insecticidal protein, and this protein present in bacteria is an inactive protoxin, but as soon as inject the inactive form due to alkaline pH of gut and converted into active form of toxin and bind to the surface of midgut and create pores that cause swelling and eventually death of the insect. Then pest resistant plants, nematode like Melodigina incognita infects the roots of the tobacco plants and causes reduction in yield. The RNAi is present in all eukaryotic organisms as cellular defense by silencing of specific mRNA due to complementary dsRNA molecule that binds to and prevent translation. Then biotechnical use in medicine, rDNA technology process have made immense impact in the area of healthcare. At present, about 30 recombinant therapeutics have been approved for human use for the world over. Genetically engineered insulin. Insulin was earlier extracted from pancreas of slaughtered cattle and pigs, but insulin from these sources developed allergy or different types of reaction to the foreign protein. Then insulin contained two short polypeptide chain A and chain B, that are linked together by disulfide bridges. Then in humans, insulin is synthesized as hormone which contain an extra stretch called C-peptide, which is absent in mature insulin. Then the main challenge for the production of insulin using rDNA technique was getting insulin assembled in the mature form. Then the American company Eli Lilly in 1983 prepared two DNA sequences corresponding to A and B chain of human insulin and introduced them in plasmids of E. coli to produce insulin chain. Chain A and chain B were produced separately, extracted and combined by creating disulfide bonds to human insulin. Then gene therapy, the correction of gene defect involves delivery of normal human gene into individual or embryo to take over the function or compensate for non-functional gene. The first clinical gene therapy was done in 1990 to a 4-year-old girl with ADA deficiency. This disorder is caused due to the deletion of the gene for adenosine deaminase that is essential for immune system. Then in gene therapy method, lymphocytes from the blood of the patient are grown in culture medium outside the body, and a functional ADA cDNA is then introduced into these lymphocytes and returned to patient. In this method, periodic infusion of such genetically engineered lymphocytes is needed, and if gene isolated from bone marrow cells producing ADA is introduced into cell at early embryonic stage, it comes as permanent cure. Then molecular diagnosis, PCR, ELISA, and ELISA is the enzyme-linked immunosorbent assay. And symptoms of disease appear only when the concentration of pathogen get increased significantly. Then ELISA is based on principle of antigen-antibody interaction. Transgenic animals that have the DNA manipulated to process and express a foreign gene are known as transgenic animals. Common reasons for development of transgenic animals: normal physiology in development, study of the disease, biological products, vaccine safety, chemical safety test. Then ethical issues. The Indian government has set up an organization as GEAC. It names Genetic Engineering Approval Committee, which will make decisions regarding validity of GM research and safety of introducing GM organisms for public services. Biopatent. A patent is the right granted by government to an inventor to prevent other from making commercial use of his invention. Biopiracy. It is used to refer the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned with compensatory payment. Several attempts have also been made to patent use products and processes based on Indian traditional herbal medicines like turmeric and neem. So this was for today's video. Hope you liked it and understood it. Thank you for watching my video.